Welcome back to the Deep Dive. Today, we are taking a massive leap forward into a technology that is fundamentally rewriting how we understand anatomy. Spatial transcriptomics, or ST.
Yeah, it really is. This is the breakthrough that's starting to unlock the deep secrets of tissue biology.
And for decades, biological researchers faced this really cruel trade-off, didn't they?
They absolutely did. You could either measure, everything molecularly, or you could measure precisely where a few things were located, but you really couldn't do both at the same time.
Okay, let's unpack that historical dilemma a bit. So on one side, you had these comprehensive omics tools, right? Like RNA sequencing.
Mm-hmm.
These gave you the full parts list they could quantify, like every single RNA transcript in a sample, a huge amount of data.
Huge amount, yeah. But, and this is the key, but to do that, you had to homogenize the tissue. You basically ground it up.
And in doing that, you just obliterated all the spatial context, all the where.
Exactly. You knew what was present, but you had absolutely no idea where it was. Imagine having the full library of components for a car, but the assembly manual is just shredded confetti.
Okay. And then on the other side, you had the targeted tools, things like advanced staining or in situ hybridization.
Right, and those were fantastic for localization. They could show you exactly where a specific molecule was sitting within an intact cell or in the tissue structure, really precise where.
And here's the other side of that coin. Those tools only let you look at maybe, 5 or 10 specific molecules at once?
If you were lucky, yeah. Maybe a few more with fancy microscopy, but nowhere near the whole picture. It was like confirming the existence of, a few key screws in the car, but completely missing the overall engine design or the chassis structure.
So spatial transcriptomics comes along and just eliminates that trade-off.
Pretty much, yeah. The whole mission of ST really is to allow researchers to quantify RNAs across the entire transcriptome, thousands and thousands of genes, all within the context of an intact tissue section.
Wow. So you finally get the parts list and the fully assembled blueprint simultaneously.
That's the goal. And understanding this massive technological pivot, well, that's our deep dive today.
That sounds genuinely revolutionary. So where do we start? What are the sort of immediate impacts? What are the core biological mysteries ST is now uniquely equipped to solve?
Well, I think ST is best suited right now to answering three major classes of questions in tissue biology. And they kind of build on each other, moving from simple cataloging up to dynamic signaling.
Okay, let's take the first one. Just the basic sense is figuring out who is actually present in the tissue.
Right. That's cell type composition. ST lets us map out which specific cell types are present and, crucially, how they are spatially distributed across that tissue section. Where are they located?
How do they know which cell is which type, though?
Well, that's often achieved computationally. You typically take these really rich cell type definitions that have been derived from single-cell RNA sequencing another powerful tech, and you essentially map those known signatures onto the ST coordinates.
So instead of just looking at a slide and saying, okay, that's a patch of cancer tissue, you can actually map it out. Like, here are the aggressive tumor cells clustering. Oh, look, here are the T cells that have infiltrated right next to them, and over there are the structural fibroblast cells.
Precisely that level of detail. And this has very quickly become the technology's most utilized application. It's foundational.
I bet.
Yeah, researchers have already used ST to create these incredibly detailed compositional atlases for, all sorts of tissues: the nervous system, human kidney, the heart, tests, lung. You name it. You are literally generating high-resolution architectural maps of life.
Okay, so once you know the composition and the location, the who and the where, the next logical question moves towards their relationships.
Yeah.
How do they relate spatially?
Exactly. That's the second big question. Cellular interactions, but focusing specifically on proximity. This helps define the rules of spatial covariation. Are cell type A and cell type B found sitting next to each other more often than you'd just expect by random chance?
Any cool examples of that?
Yeah, there was a neat study on the mouse visual cortex. They found that specific subtypes of inhibitory neurons actually preferred to cluster together spatially. They weren't just randomly sprinkled throughout the tissue. There were rules to their organization.
And that spatial proximity must be fundamental information, especially when you're looking at disease states.
Oh, absolutely critical. Proximity analysis is being directly applied to pathology right now. People are using it to map gene expression patterns, like right at the very edge of amyloid plaques in Alzheimer's disease tissue.
Right.
Or to define the specific organization of immune cells linked to how tissue responds pathologically after, say, a traumatic brain injury. It tells you who's talking to whom locally.
Okay, so that leads us to the third And maybe the most sophisticated question, not just that they're next to each other, but what are they actually, exchanging? What signals are passing between them?
That's right. That's molecular interactions or communication. Because ST gives us data on virtually all genes, we can start to define potential signaling pathways. We look for active ligand-receptor pairs among cells that are spatially adjacent or nearby.
Explain ligand-receptor pairs quickly.
Sure, ligands are basically the signaling molecules, the messages that cells send out. And receptors are the specific receiving stations on other cells designed to pick up those messages.
Okay, so ST lets us essentially see both the message being sent and the receiver picking it up right there in the tissue space.
Exactly. You can visually confirm a potential communication line. Like you mentioned earlier, maybe a tumor cell sending out a specific help me grow signal and an adjacent blood vessel cell expressing the exact receptor needed to pick up that signal and respond.
That's powerful.
It's crucial for understanding what we call cell non-autonomous phenomena interactions that happen between different cells. Things like how solid tumors chat with their surrounding microenvironment, how developmental gradients get set up in an embryo, or even how immune cells coordinate their attack patterns within infected tissue.
Composition, proximity, communication. Okay, 3 really powerful layers of information, all unlocked simply by keeping that spatial context intact. Amazing. Now let's shift gears a bit and look under the hood at the engineering. Our sources make it clear that ST didn't just spring from one invention, right? It seems to evolve from 2 parallel, pretty high-tech tracks. How do we distinguish these 2 families of technology?
Yeah, that's a good way to put it. We generally classify them into 2 major families, primarily based on how they read out the information. We talk about sequencing-based ST or SST and imaging-based ST-IST.
Okay, SST. I'm picturing that coming more from the traditional genomics lab, the sequencing world.
That's pretty accurate, yeah. SST really leverages all the amazing advances we've seen in massively parallel DNA sequencing and molecular barcoding, very similar actually to the tricks used in single-cell sequencing.
And the core mechanism is?
Molecular capture. The basic idea is you use a special surface, like a microscope slide, that's coated with DNA primers. But here's the key. Each primer is tagged with a unique location-identifying barcode, like a tiny little zip code.
Okay.
Then you place the tissue section on top of this surface. The RNA molecules diffuse out of the cells, get captured by these primers, and become tagged with that location-specific barcode. Then you collect all that RNA, sequence it, and the barcode tells you where each RNA molecule originally came from.
Got it. So SST is kind of a catch-and-sequence approach. You capture the RNA, tag its address, then read it all out digitally later to build the map. What about IST then?
IST, on the other hand, really grew out of the microscopy and cellular imaging world. Imaging-based IST, aims to keep the RNA in situ, meaning right in its original spot within the cell or tissue.
So no capturing onto a slide.
Generally not, no. It detects the mRNA transcripts directly using advanced fluorescence techniques. Things like specialized versions of in situ hybridization or even performing sequencing reactions right there on the slide while it's under a microscope, you're seeing the RNA in place.
That makes the distinction really clear. It's capture versus direct detection. Okay, let's dive a bit deeper then into sequencing-based ST first. The way they actually create those surfaces with the tiny barcoded pixels. That sounds incredibly clever. Lots of different engineering tricks.
Oh yeah, there's a lot of ingenuity there. But the central idea for all SST methods is creating that spatially indexed surface. We can sort of simplify the engineering approaches into maybe 2 main categories, often differing by the resolution they achieve. The first category uses deterministic printing.
Like a very, very precise, high-tech robotic printer.
Exactly like that. Methods like the popular Visium platform use automated microarrayers. These are essentially robots that very precisely print unique location barcodes onto defined spots on the slide surface.
How big are these spots?
They're relatively large in cellular terms, maybe 50 to 200 micrometers across. So each spot captures RNA from a small neighborhood of cells, not usually a single cell. It's very robust, reliable, but the resolution isn't super high.
Okay, so that's one way. What's the second category? That must be how they achieve much higher resolution, maybe getting down closer to single-cell scale or even smaller.
That's right. That's what we could call the self-assembled or high-density category. This involves a range of technologies that create these massive arrays of incredibly small indexing structures. Sometimes they achieve subcellular resolution down to the nanometer scale in some cases.
How do they make such tiny things?
Well, there are different tricks. One way is by randomly depositing millions of tiny pre-barcoded beads onto the surface, like with SlideSec. Another way uses specialized DNA amplification processes, things like rolling circle amplification or RCA, to generate tiny, dense clusters of barcoded DNA. These are sometimes called nanoballs or polonies.
Nanoballs? Wow.
Yeah, they can be incredibly small, like 200 nanometers. Because they're often generated randomly, you then use imaging or other methods to figure out the precise location of each bead or nanoball computationally after the experiment. There are also microfluidic approaches, like D/T-Sec, that use tiny channels to deliver barcodes.
Okay, so you either print defined, fairly large spots, or you create millions of tiny, dense, often randomly placed points and figure out their addresses later. But either way, you end up with gene expression data tied to specific coordinates.
That's the core idea. But, regardless of the specific method used, if we want to compare these different SST technologies or even results from the same tech across different labs or tissues, we desperately need good quality control.
Makes sense. What are the key things to measure?
The two most critical parameters for SST performance are, first, mRNA capture sensitivity, basically. How efficiently does it actually grab the RNA molecules from the tissue per unit area? Are you detecting a lot of the RNA that's there, or just a fraction?
And the second?
The second is spatial accuracy. This is really challenged by something called lateral diffusion.
Ah, right. That's when the RNA molecule kind of floats sideways away from its original spot before it gets captured by a barcode, blurring the map.
Exactly. It degrades the precision of your spatial map. And because both sensitivity and diffusion can change wildly depending on the tissue itself, think about things like how much RNase enzyme is present, which chews up RNA, or how densely packed the cells are the field is. Well, it's practically screaming for standardization. It's hard to compare apples to oranges right now.
So what are researchers suggesting? How do you standardize this? Do we need, reference points?
Yes, exactly. We need widely adopted reference tissues that have known, reliable, and complex enough histology that everyone can use them as a benchmark.
What kind of tissues work for that?
Well, two main examples are often proposed in the literature. First is the adult mouse olfactory bulb. It has these 5 very clearly defined molecular layers with well-known gene markers for each layer, very structured. The second is the embryonic day 12 to 14 mouse embryo, specifically looking at a sagittal section that includes the developing eye. The eye provides these really distinct, highly reproducible, symmetrical histological layers.
That makes sense. You have clear landmarks.
Right. If every lab developing or using an SST technology ran these same reference samples using agreed-upon metrics, we could finally start speaking the same language regarding performance. We could properly benchmark sensitivity against, say, single-molecule FIS references and quantify that blurriness from diffusion by measuring feature sharpness.
Standardization sounds crucial.
Ah.
Okay, let's pivot now to the other family, imaging-based ST or IST. You mentioned the traditional hurdle for fluorescence imaging is the spectral limit. You can only reliably distinguish maybe 5, perhaps 10 different colors at once. How on earth did IST get around that to read out potentially thousands of different genes?
Yeah, that was the fundamental challenge. And the key strategy they employed is essentially using time to overcome the limitation of colors. They achieved this really high multiplexing by using multiple sequential imaging rounds and relying on some very clever combinatorial coding strategies.
Okay, multiple rounds. But how is the RNA actually labeled or detected in the first place in these methods? What are they actually imaging?
They generally use 3 main ways to visualize the RNA. First, there's direct probe-based detection. This is basically an enhanced version of a classic technique called single-molecule FESH, or SMFesh. You design many short fluorescent probes that all bind along the length of a single RNA molecule. Together, they create a bright, specific spot you can see under the microscope.
Okay, lots of little lights make one bright spot per RNA. What else?
Second, they sometimes use enzymatically assisted methods. Here, they use molecular biology tricks, often involving enzymes like polymerases doing something called rolling circle amplification, RCA, right there in the tissue. This creates a much, much brighter amplicon, a big ball of DNA copied from the RNA target, which really boosts the signal-to-noise ratio, makes faint signals easier to see.
Brighter signal means easier detection. Got it. And the third way?
The third approach is direct enzymatic sequencing. This actually tries to perform sequencing reactions, figuring out the RNA sequence base by base directly within the tissue sample. Conceptually, this is maybe the most similar to SST because it can be untargeted, potentially sequencing any RNA present. However, it's often limited by a fairly low conversion rate of the original RNA into the final signal you can detect. It's technically quite challenging.
Okay, so you've got ways to make the RNA targets light up. Now for the magic trick, as it were, the
Right.
multiplexing. Once the RNA is labeled, how do they actually read out thousands of different gene types? I know you mentioned sequential rounds. There's a simple approach of labeling some, imaging, stripping off the probes, labeling others. But that only gets you so far, right? Maybe 100 genes before the sample falls apart.
Exactly. That sequential stripping approach has scalability limits due to sample degradation. The real breakthrough for high multiplexing is combinatorial multiplexing. This is different. Instead of labeling a unique set of genes in each round and then trying to remove those labels completely, here, each individual RNA molecule is interrogated over multiple imaging rounds.
Okay. How does that work?
Its identity isn't based on just one color in one round. It's decoded based on the combination, essentially a binary code of images or rounds in which it was detected versus not detected.
Ah, I see. So if a specific RNA molecule lights up, say in round 2 and round 5, but it's dark in rounds 1, 3, and 4, that specific pattern, like 0101, is its unique identifier corresponding to a specific gene in your codebook.
Precisely. And the beauty is that the potential codebook size scales exponentially. With n imaging rounds, you can potentially encode 2 to the power of n different gene identities. So even with just, say, 16 rounds, you have 216, which is over 65,000 unique codes, more than enough for the whole transcriptome.
That's incredibly efficient.
It is. But here's another crucial detail that makes methods like MERFISH work so well. They don't just use simple binary codes. They use mathematically designed Hamming distance-corrected, error-robust codebooks.
Okay, what does that mean in simple terms?
Think of it like a super robust ID badge. It has enough redundancy built into the code itself that even if there's a mistake in one of the imaging rounds, maybe a spot doesn't light up when it should have, or a wrong spot appears, the system can often still uniquely identify the molecule and correct the error. It can tell that 01011 was supposed to be 01001. This dramatically improves the accuracy because inevitably, things sometimes go wrong during one of those many imaging rounds.
That makes the whole system incredibly resilient to noise and error. But surely this massive detection effort, trying to image potentially millions of molecules over many rounds, must run into some physical limitations.
Oh, it absolutely does. The primary bottleneck, especially as you push for higher and higher numbers of genes or just denser tissues, is often referred to as the crowding problem.
Too many spots too close together.
Exactly. As you try to image more and more individual RNA molecules within a packed cell or tissue, the fluorescent spots start to overlap. They get too close together, which makes it really hard for the imaging system and the algorithms to accurately distinguish them and decode their combinatorial codes.
So how do they deal with that crowding?
Well, sometimes researchers have to compensate by using other techniques, like expansion microscopy, where you physically swell the tissue sample, making it bigger to space things out. But that, of course, comes at the cost of throughput. You can't process as many samples if you have to expand them all first. It's a trade-off.
Okay, so we have these two incredibly powerful paths, barcoded capture with sequencing, SST, and combinatorially coded imaging, IST. Both are giving us just unprecedented spatial molecular data. But our sources strongly suggest this is really just the dawn of contextual genomics. What comes next? Where is this heading beyond basic ST?
Yeah, I think that's right. ST is foundational, but we're already seeing innovation pushing along 3 major trajectories. First, expanding the types of molecular modalities we can measure spatially. Second, relentlessly pushing for higher resolution. And third, adding the dimension of time, moving towards what some call 4D biology.
Okay, let's start with expanding modalities, going beyond just standard messenger RNA.
Absolutely. The same core principles, spatial barcoding, high-resolution imaging, can theoretically be applied to almost anything within the cell or tissue. We'll definitely see, and are starting to see, spatial profiling of genomic variation, things like mapping specific mutations in cancer tissues spatially.
And epigenetics.
Yes, epigenomic regulation, mapping where the chromatin is open and accessible for transcription, for instance. And a huge area is integrating proteomics.
Measuring proteins spatially?
Several ways. You can attach DNA barcodes to affinity regions, like antibodies or aptamers that bind specific proteins, and then use spatial capture methods similar to SST. Or you can use clever proximity-based enzymatic reactions that generate a signal only when 2 specific proteins come very close together, allowing you to map protein interactions in situ.
Wow. So the dream is a unified map showing DNA status, RNA expression levels, and protein locations and interactions all in the same tissue section.
That's the vision, a truly multi-omic spatial atlas.
Which leads directly to the next point, resolution. We've talked about spot sizes and crowding. How do we get from mapping general tissue regions or cell neighborhoods right down to precise subcellular localization?
That's a major frontier. Currently, as we discussed, most SST methods that don't have single-cell resolution require computational methods deconvolution to estimate which cell type contributed which RNA molecules to a mixed pixel. That gets easier as pixel size approaches cell size, of course.
And for IST?
For IST, the challenge is often computational, too accurately segmenting the image to assign individually detected transcripts back to their parent cells. But the future goal is really about precise subcellular localization, knowing not just which cell molecule is in, but which organelle within that cell.
So knowing if an RNA is in the nucleus versus the cytoplasm, or if a protein is on the cell membrane versus inside mitochondria.
Exactly. The goal is to develop technologies that can precisely colocalize different types of molecules, maybe even pushing beyond the diffraction limit of light using super-resolution techniques to see things like transcription factors binding to specific DNA sites or biomolecules accumulating in specific subcellular compartments. True molecular cartography within the cell.
And finally, the dimension that's always the hardest in biology.
Yeah.
Moving from a 3D snapshot, however detailed, to a 4D living map of biology. How do we observe dynamics?
This is perhaps the most exciting and maybe most challenging frontier, observing cellular dynamics spatially. One really innovative approach is using something called molecular recording.
Molecular recording, what's that?
This involves using synthetic biology tools, often based on CRISPR gene editing systems, to actually make the cell itself encode its own history, things like its lineage, who its ancestors were, or what signaling pathways have been active directly into its own genomic DNA sequence over time.
Whoa. So the cell itself is keeping like a running historical ledger of its life experiences written into its DNA.
Kind of, yeah. And then at the end point, you can come in with spatial sequencing methods to read out both the final state of the cell and this recorded history, all in its spatial context.
That sounds incredibly powerful for things like developmental biology, tracking how tissues form.
Exactly. Or for understanding tumor evolution and clonal heterogeneity, how different subpopulations of cancer cells arise and spread. Another way we're starting to get temporal information is through clever computational inference.
Using the spatial data itself to infer time?
Sort of. By leveraging subtle spatial information, for example, precisely measuring whether a specific RNA molecule is located primarily in the nucleus, which might indicate very recent transcription, versus finding it mostly out in the cytoplasm, suggesting expression that happened some time ago, you can start to infer temporal dynamics, even from a single snapshot. It helps move us beyond just a static picture.
This whole deep dive really highlights just how pivotal spatial transcriptomics is. It feels like it genuinely bridges that long-standing gap between comprehensive omics data and precise tissue context, giving us these incredibly high-resolution maps of cell composition, their interactions, and even their communication.
It really is transformative, but it's important to remember, and the sources are quite clear on this, that to truly accelerate progress and make this technology robust and widespread, the field absolutely must resolve two major challenges, like right now.
Right. You mentioned the urgent need for standards earlier.
Yes, it bears repeating. First, the immediate and widespread adoption of standardized reference tissues like that mouse olfactory bulb or the embryonic eye we talked about is absolutely necessary. We need them for rigorous benchmarking, for comparing performance fairly across all the different platforms and labs.
Had everyone on the same page regarding quality.
Exactly. And second, alongside that, we desperately need the development and adoption of generalizable open source image processing pipelines and standardized data formats. The amount of complex imaging and sequencing data these technologies produce is staggering, and right now, analyzing it is often a bespoke, lab-specific bottleneck. We need common tools.
So if we can tackle these standardization and data handling issues, it really clears the path for this future of contextual multi-omic dynamic genomics.
It does. We really do now have a clear technological trajectory emerging for generating these true 4D maps of biology integrating space, molecules, and time.
Which leads us to our final thought for you, the listener. We leave you with this provocative question. If you could choose just one biological system, or maybe one particular disease, any biological mystery you care about, and apply these advanced 4D mapping tools with their full potential for spatial, molecular, and temporal resolution, Which biological question would you try to solve first, and why? What map would you want to create?
Think about that. What secrets would you want to uncover?
We'll catch you on the next deep dive.